This is part 3 of my lab practicum. So I'm going to be showing you how to do the phenol red broth. So the first thing I'm going to do is get my Bacto incinerator in my inoculation loop. And I'm going to incinerate it to sterilize it for 10 seconds. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So now that this is sterile, I'm going to take my test tube of culture. I'm going to open it, dip my loop in now that I've waited 3 seconds for it to cool, cap it, put it back in my test tube rack. Now I'm going to take my sterile broth culture, open it, place the loop inside and give it a little stir, making sure to not get any bubbles. Now I'm going to recap the broth culture, and I'm going to put it in my test tube rack until I'm done. I'm now going to Bacto incinerate my loop for 10 seconds. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is sterile, so I can now put it down again. So now I can take the broth culture that I just did and put it in the incubator. After I incubate it, I should be able to tell the results. I should be able to tell like acid production and if there's any fermentation in this. So give me a minute and I'm going to pause this video so that way I can set up to show you the next part of the lab practicum. Okay, so welcome back to my lab practicum. Now I'm going to do a TSI slant. So the first thing I'm going to want to do is get my Bacto incinerator again. This time I'm going to use the inoculation loop and I'm going to sterilize that for 10 seconds. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Wait 3 seconds to let it cool. Again, I'm going to get my culture here. I'm going to open it, dip my loop, pull it out, cap. This now goes back in my test tube rack. This would be my slanted auger, but I did not put Jello in it this time, as explained in other videos. So I would cap it like I would. I would put it into the slant, pull it out, cap it. This now will be incubated for, I don't know, probably 24 to 48 hours. And then I should be able to tell the results of like fermentation and stuff, especially like lactose fermentation. So let's say we put that in the incubator. This is going to go back in our Bacto incinerator. 10 seconds to sterilize. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, now that this is done, we can set it down. Now for the phenol red broth as well as the TSI slant, once you're done, you can put them all into the biohazardous waste. I forgot to say that. So whenever you get rid of something that isn't glass, always goes in your biohazardous waste. So give me a minute to set up and I will show you the next one. Okay, so now I'm going to show you how to do a gelatin test. So again, our handy dandy Bacto incinerator will come out into play. I'm going to take my wire again, sterilize it for 10 seconds. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Wait 3 seconds to let it cool. Now I'm going to take my culture again, open it, dip my loop down in, cap it. Now that this is capped, goes back in our test tube rack. Now I'm going to take our gelatin filled tube and I'm going to stab down into the gelatin and pull out. And this would get my culture inside the gelatin, so that way we can see how far it grows or will not grow during our test. And then I'm going to cap it and incubate it. Now, incubating it will turn the Jell-O into liquid. So once I'm done, I need to put it on ice for 5 minutes, and then I'll be able to tell the results. Now, if it's still liquid, it's a positive test result, but if it's solid, that means it's negative. After we're done with our test, this is always discarded in our biohazardous waste. So, there it goes in our biohazardous waste. My wire, I always put back in the back to the incinerator. 10 seconds again. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Now I can set this down. So now I'm ready to show you the next part of the lab. So let me get that set up. Hi, welcome to my next part of the lab where I'm going to show you the catalyst test. So for this, you would have an already prepared slant stock culture. So I'm going to drop a catalyst reactant in it. So I'm going to pretend this is catalyst reactant, and you put a few drops in. And then I can cap it. I would now incubate it so that I can see the results of my test. If bubbles form in my test, that means a catalyst is present. If there's no bubbles, that means my catalyst is absent. Once I, again, am done with this, this goes in the biohazardous waste. So here I am discarding it. And let me show you the next part of the lab practicum. Okay, next on lab practicum is the EMB, which is simply a repeat of the streak plate with a different auger. So I have a different auger in here. I labeled the bottom, did my 3 lines, 3 lines, 3 lines, intercrossing each. So now, if I did this, set this up right, and it's got my auger in it, I'm going to show you how you would put your culture in. So you would take your culture, like before, make sure it's the right one, take your back to incinerator, end your loop, and incinerator, 10 seconds, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is ready to go after 3 seconds, so 1, 2, 3. I am now going to dip into my culture with my loop. So I'm just going to dip right in, cap it, put it back in the test tube rack. Then I'm going to clamshell and from left to right go over the 3 lines in my test tube. Not my test tube, my Petri dish. Then what do I do with this? Back to incinerate again, like I did in my streak plate. So 10 seconds. 10 seconds go by. 3 seconds, cool. Clamshell again. Run back over your lines. Another set of 3. Close. Back to incinerate. 10 seconds. 10 seconds. 3 seconds, cool. Run back over. Another set of lines. Pulling your already put in culture through. You're not going to go back in and dip for more culture. It would look like this. Now this can be incubated, and you should be able to see the growth, the colonies, colors, anything that you would see on a normal streak plate dish. So once you're done with this, like any streak plate or anything like I mentioned that could be biohazardous, would go in our biohazard waste. So now I'm going to set up and show you how to do the next part of the lab practicum. Okay, so now I'm going to show you how to do your minimal salt auger. Again, just like your last one, this is going to be just like your streak plate, but with salt auger. So, taking my loop, back to incinerate, 10 seconds. Gonna take my culture here, open it, dip in after I waited 3 seconds to let my loop cool. Otherwise, I would kill all the bacterial cells I'm testing. Now, I've already labeled from before. So I showed you I labeled my name. And I labeled unknown because I don't know what bacterial I'm testing. I did my 3 lines of 3 intercrossing each other. So now I'm going to clamshell from left to right, run over 3 lines, close, rebacto incinerate, 10 seconds, wait 3, clamshell, pull the already used culture from the other lines through 3 more lines, close, rebacto incinerate my loop, 10 seconds, wait 3 to cool it off, pull back through the remaining 3 lines. Now this can go in the incubation for probably 24 to 48 hours, and by then, like most streak plates, you'll be able to tell how many colonies, the colors, how the growth was, so on and so forth. So now, once I am done, I can put this in the biohazardous waste as mentioned before. So with that, I'm going to move on to the very next part in our lab practicum. So now I'm going to show you how to do the starch test, which again is similar, but with different agar. So, like before, bacto incinerate your loop, 3 seconds, let it cool. You incinerated for what? 10 seconds, remember. Now I'm going to take my culture again, open, dip in, pull out, cap, test tube rack. Okay, what do? I'll be clamshelling, pulling through the first set of 3 lines I want to do. Okay. Rebacto incinerate, 10 seconds, wait 3. Clamshell, pull your culture through. Don't dip back in. Don't go back in that test tube. Bacto incinerator again, 10 seconds, wait 3. Pull through your remaining 3 lines. Now, unlike the other ones, we're not done here. So I'm going to bacto incinerate my loop so I can put it down. 10 seconds, 3 to cool off, and I'm setting that down. For this one, you would need iodine, which I don't have the card for, so I'm going to imagine this is our iodine. I'm going to put a few drops. I'm going to flood the petri dish with it, the top of it. And then I'm going to put it in my incubation after 5 minutes. Oh, no. I'm not going to incubate it. I'm going to put it over a weight sheet after 5 minutes. And that should help you see the growth, the colonies, colors, and so on and so forth. But once I'm done with this dish gets put in the biohazardous waste. Next is blood agar, and we do it the exact same way, except without the iodine. So we're going to bacto incinerate 10 seconds. Dip into our culture after 3 seconds of letting it cool. Clamshell, pull through 3 lines. Close. Oops, I'm sorry. I should never put a test tube down on the table. That is my fault. This should have gone into the test tube rack. Always remember, don't put your test tubes on your table. That was my fault. So I'm going to rebacto incinerate. Okay, now I'm going to pull the remaining culture through the 3 lines. Close. Rebacto incinerate. 10 seconds. Wait 3. Pull back through the remaining lines. Now, with the blood-rich agar and your culture, put it in the incubation for 24 to 48 hours. And again, with normal streak plates, you'll be able to see colonies, growth, so on and so forth. So now I'm going to pause and get ready for our next part of our lab practicum. So the very next thing on our lab is to do the motility test. So this, I will have a tube full of the agar or whatever we were using. I would I would incinerate my wire, 10 seconds, just like our loop, wait 3. I would take our culture, dip my wire in, enough to get some culture, cap, put back in test tube wrap. That's what I gotta remember, right? This I would open, stab right into the auger or whatever we use for motility testing, cap it. Now we can incubate and record. Now if it's going to grow, it's going to continue to grow down. If there's no growth, there's no growth. This helps us to record what kind of growth is happening. So after this, I would dispose of this in biohazardous waste. Put this in my back-down incinerator. 10 seconds, 3 to cool down. Can put it back down on my table. Last thing I want to do is clean my area. So I need to get my good old germicide out here. Let me get it. Here it is. I'm going to spray down my table. Use my paper towels, wipe it all up. I'm going to make sure that all my stuff is disposed of properly. Slides go in sharps containers. Anything other biohazardous will go in your biohazardous waste. So when I'm done cleaning up, I'm going to put my gloves and I'm going to put them in the biohazardous waste just to be safe. So that way nothing can get contaminated from my gloves. I will take my gloves off by pulling through the hand, not to touch the skin, fold it up, crinkle it in my hand, fold it underneath, and pull it up. So that way nothing on the glove that I touched is touching me. Only what I touched on the inside of my glove. And I would throw these in the biohazardous waste. Everything is clean, and I would put my lab jacket up. And that is the end of my lab practicum.